You guys hear me?
Yes.
Awesome. All right, give me one more second. See if I can paste. I forgot to paste the link on Brightspace, so let me get that going. I think I'm gonna probably either I can draw on paper and then I'll upload the pictures afterwards, or I'll probably or I can try to open up the slides and we'll kind of talk through them a little bit. If you guys want to start getting your questions ready, if not, I'll start going through the ones I got from the group stuff. I think we'll give them about another 5 minutes. Hopefully people show up. Ooh, give me a second. I'm gonna get a napkin real quick. I'm dying. Hey, buddy. So I feel that a lot of the questions I had were about the pioneering factors. So I think I'm gonna start with those guys. All right, cool. It's gonna be a small group today. All right, let me share my screen. I'll try doing the share screen. If not, I'll jump over to the paper stuff just because I feel a little more comfortable with it. So let's start here. And what we're looking at here is about, like I said, like almost at the very, very beginning of the story when it comes to gene expression, it's about how accessible that DNA really is, right? If, let's say we imagine we just got out of cell division, right? Your DNA would have been super compact in the chromosomes, basically stored away. And now it's time to start opening it up again during the interphase, trying to start working with the DNA itself. So in order to do that, you basically need to get it off the histones. So here in blue, we're basically drawing the histone complex. So in here, for this part where the histone complex, the main thing is that I do expect you guys to know the 4 parts on the inside, right? H2A, H2B, H3, and H4. That's where your DNA is going to be coding around it. And then H1 is the one that kind of hooks up at the outside between the loops. You have 8 of these H2 to H4, and then you only use one H1 to make that complex happen. Now we're super condensed, our DNA is super compact. There, I want you guys to know your pioneering factors. So this one, I have not gone into the details about what exact piece of DNA they recognize or what they grab onto to pick a target, which I'm okay with that, because I just kind of want you guys to know that there's different pioneering factors and that each one of them recognizes different sequences. That's as far as I want you guys to go get into it. And what basically happens is, let's say you had your kidney cells, and then you had, let's say, what was the ones you did an example? Your kidney and your lung cells, right? Your kidney cells will express certain pioneering factors that open up the genes involved for the kidney, while your lung cells will express pioneering factors that are genes for the lungs. So there's two different, there's multiple types of pioneering factors that recognize their own sequences. I'm not going to give you a specific one to be like, hey, pioneering factor likes sequence, blah, 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 blah. If I ask you for this kind of question in the midterm, I'm going to be asking you things like, hey, we have a pioneering factor that recognizes, knowing that it recognizes in a specific tissue and it'll work at that spot. The next step for this one, we're going to talk about those pioneering factors bind to the DNA. And the difference between a pioneering factor and a regular enhancer is your pioneering factor will actually grab onto DNA despite histones being bound there. So if you imagine this is my rough example. Sorry, trying to get some rope. There we go. So you have your DNA, your DNA. My hand's basically the 4 histones, your DNA like this. Now, a pioneering factor can go, and even though this DNA is interacting with the histone, it can still go and recognize the sequence that it likes, bind to it. A regular transcription factor, let's say the blue one is my transcription factor, this one cannot recognize the DNA if it's bound onto the histones. So that's the only thing that sets these two apart, right? So your enhancer, your pioneering factor, it has a sequence it likes. It can bind it despite the histone being there. Your transcription factor that binds to enhancers, it has a sequence that it likes, but it can only do so if that sequence is not open for it to interact with. If it's wrapped up, it's basically inaccessible. So those are 2 things that set those apart. Now let's go back to talking about our pioneering factor. This pioneering factor interacts with this sequence that it likes, is basically scanning around until it finds that perfect sequence, right? Once it finds that perfect sequence, here you're going to bring in enzymes that help modify the histones. So either you can modify the histone tails. Remember, we talked about 3 different types. For this, my super big emphasis, or the ones that I really, really want you guys to know, are these 3 that we talked about in the class. And all you need to know is know them at this depth, right? Acetylation and phosphorylation, both of those lead to gene expression. And that's because those modifications end up having the DNA basically fall off the histone or release from the histone, right? The methylation, on the other hand, getting the methylation to happen actually makes the DNA coil tighter, and that's how you get that super fine wrapping like this, right? And you have multiple histones sticking next to each other. So know the 3 ways that you can modify them. Just know how to group them, kind of how we did in that in-class exercise. Then the only one that I expect you to know in greater detail is going to be the acetylation, right? The other 2 work the same way. They have their own specific enzymes, but I want to talk about how that interaction is reversible. So for this one, I do want you to know this first one here called a histone acetyltransferase. So what's going to happen here is your pioneering factor shows up. This is going to bring in your histone acetyltransferase. So that's a different enzyme. You can tell kind of a little bit by the ending, calling it an ACE. So transferase, that's the enzyme that adds the acetyl groups to it. So as you can imagine, pioneering factor, you bring in that extra enzyme that modifies the histone, right? And you can see adding acetylation leads towards gene expression, right? So now here you have your condensed DNA at the top. Your pioneering factor shows up. You're going to bring in that histone acetyltransferase, that enzyme. You're going to modify the tails of the histones, which was before super tight. Now it's getting modified. It's going to start releasing it. this is going to mean that part of the DNA is going to open up. And let's say we had a region of this rope that only liked binding to that transcription factor that has the specific enhancer. Because we've moved the histones away from it. Now, remember, now our transcription factor, I should put it red like the one in the thing. Now your transcription factor has a chance to actually land there and can recognize the sequence, and you can start building your complex to get expression to happen, right? So I'm gonna stop there from going on with how expression happens, but let's go back to, we have the DNA, because we've added the acetylation, the DNA has fallen off the histone, right? On the next slide, the one I expect you guys to know, let me unbold the other one. That's the name of one type, the methyltransferase, but the one that I really want you guys to know, or the one I'm gonna emphasize in the midterm, is gonna be this one, the histone deacetylase complex. As the name kind of implies, what it does is it takes away the acetyl groups. So remember before, our histone was opened up because it had the acetyl group, it had got pushed off the DNA. Now, this complex comes in, removes the acetylations off the tails. Now it lets the DNA wrap better around the histone, and boom, we're back to having that condensed DNA. Yeah. So I expect you guys to know the pioneering factor, which helps us target the gene that we care about, right? Next, we have the hat, the histoacetyltransferase comes in, adds the acetylation, that opens up the DNA. Then after once you need the DNA to be silenced, right, you have your deacetylation happens, you remove the acetylation, your DNA coils back up, and you basically put that DNA back away. As long as you know that it's a cyclical event, that you basically have two enzymes that are controlling this, right? Either you're going to be in deacetylation or you're going to be in deacetylated. And you're going to have these two enzymes that are doing the process of moving the DNA this way or moving DNA the other way. Just like that, there's different enzymes that I could talk about for the methylation. Like here's this histone methyltransferase. For phosphorylations, there's a thing called kinases, and I think the other one called, kinase and phosphatases. They basically do that reverse role. But I want you just to know that in general, they can be switched on and off. The one that I expect you guys to know by name is this one, the H stack and the histone acetyltransferase, so the hat. As long as you guys know those two, feel really comfortable with those two, you guys should be all right. Give me 1 second. Between histones. Oh yeah, so along those lines, I will not give you a specific sequence for the pioneering factors. I could have you theorize, right? And I could say like, hey, if we have let's say we're looking at 10 different genes. 5 of them are expressed in the kidney, 5 of them are expressed in the lungs. Like, which ones would you expect to have similar pioneering factors, right? And from there, you should kind of realize that, hey, if all of them are expressed in the kidney, most likely those are going to share similar sequences for a pioneering factor. While if they're expressed in the lungs, you'd expect those genes to have very similar pioneering factors. Same thing I would say when I talk about transcription factors binding to enhancers. It's like those transcription factors are basically a way to turn multiple genes on using that same enhancer sequence. So if you need a gene to be expressed in one specific tissue, and you need multiple genes of different types being expressed, all you do is you basically have that enhancer in front of all those genes, and you can get expression in that particular tissue. So those are the two things that are going to be similar within a specific tissue, and it's going to be different for each specific tissue type that you want. How are you guys feeling about the enhancer stuff? Like the sequences, that kind of makes sense? Give me some thumbs up, thumbs down. Nice. I think in the chat, try it. There's a thumbs up thing. That way you guys don't have to. Or the little, what are they called? Smiley faces. There we go. Perfect. If I get any thumbs down, give me some specific questions, then I'll go over them. All right, we're doing good. All right, thanks, guys. Strategies. Answers. All right, so here I got a question about like how do the enhancers fit into the transcription translation story, right? In as a big picture. For this one, let me see. I think I might have to go old school and do a little bit of drawing on this guy. I got some butcher paper, y'all. This thing is isn't dope. It's like the ultimate whiteboard. So let me think. So the way to think about this story, and the way that I'm probably going to ask you when I bring it up in a midterm, is going to be one of two ways. To understand the whole thing between pioneering factors, enhancer sequences, you can either think of the exact same piece of DNA in two different cell types, right? For example, let's say I tell you, hey, we're talking about I think from the last in-class assignment, it was like, we're talking about this one gene, and we're like, how would this gene behave in two different tissue types, right? Either the lungs and the liver, if the gene's only found in the liver. The other way to look at it as well is you can say, hey, if we're only thinking about a single liver cell, how do two genes behave in that cell if one gene is normally expressed in the lung and one gene is normally expressed in the liver? You should be able to compare analyze those two promoters and enhancer sequences based on those two things, right? So I believe the in-class assignment was one gene in two different cell types. So today, just for practice sakes, I'm gonna do it backwards. What we'll do is I'm gonna talk about a single cell type, and then we're gonna talk about how two genes within that could behave if one gene was specific to the lungs and one gene was specific to the liver itself. All right, so here. Like my whiteboard? Thanks. All right. So we're going to imagine this white paper is going to be the inside of a cell. And we're going to go for the red. It's going to be our DNA that's going to be, or the gene that's going to be a lung gene, right? Can you guys see that? Is that too small? I think I might have to make it bigger. All right. Does that look better? Can you guys read it? Give me a thumbs up. Otherwise, I might have to sketch it afterwards online. But red is going to be lung, and blue is going to be liver, right? Now, if we imagine their piece of DNA where they're both sitting inside the same chromosome, so for the DNA itself, I'm going to start off, I'm just going to draw what you'd imagine, right? Let's say this long piece of DNA like that. Let's say these genes happen to be right next to each other, yeah? In this red part, let's say right here, we're going to keep it super simple because that's not the main point of the story, but let's say this was the coding region. It was all exon, there was no introns, we code the whole thing. Your lung gene is this guy right here. And on this side, we have our liver gene like that, right? So again, same thing. It's one single exon. There's your ATG. This whole thing gets made into a protein. We hit the stop. All right, so now that we're oriented, what we're going to do is here in purple, let's say this would be our sequences that are recognized by the pioneering factor. The lung, in front of it, the genes that are belong in the lung, they have the enhancers or the pioneering sequences. You can find them on their promoter. Wow. So this would be, I'm going to put L for lung, pioneer, sequence, right? On the other side, you're going to have these little sections like this, and this will be the sequences, which will be your liver pioneer sequence. Yeah. So what's going to happen there is because we're working, or let's say right now, if we're sitting inside of this is a liver cell, and we're sitting inside of the liver, what you'd find would be that the pioneering proteins that be found. So this is your liver pioneer protein, right? Because we're in the liver pioneering factors would be present. This protein would be able to go bind to this DNA, like so. Because it doesn't have the same sequence and it doesn't recognize the sequences for the ones inside the lung, this pioneering factor will not bind to these sequences, right? So at this point, what's going to happen is, again, let's stick with only my favorite, or the ones that I really want you guys to know for your midterm, is that if we have the liver pioneering factor, it's going to show up, it's going to bind to this DNA. So the sequences it recognizes, that's going to be your step one. The next step is, this is going to recruit your histone acetyltransferase, right? Now, this one's going to be its own little enzyme, like so. And let's imagine that it carries around with it those acetyl groups. It's not exactly how it does it. It glues them in the spot, but just for cartoon's sake, we're imagining that our histone acetyltransferase is carrying a little acetyl group. And what's going to happen is that when this pioneering factor interacts with your histone acetyltransferase, now you're going to have the acetyl groups getting attached to your histones, right? That means that only on this side, you're going to go from a coiled DNA to an open DNA. On this side, because we're inside of the liver, there's only liver pioneering factors, the lungs are not going to get opened up at all, because since there's no pioneering factors, there's no chance for a histone acetyltransferase to come in and help. So what's going to happen is, this DNA at the end of the day is still going to be coiled around the histones, right? You see that? Yeah, hold on. Coiled on the histones. While on this side, this DNA will actually be free like this. Your histones might be on the outer edges like that, but the region where you have the promoter sequences and where you have your coding region, all of that is actually going to be free. And that's the whole difference of how, at this very, very early stage, about how you're going to find the DNA, it's actually being set up by the pioneering factors. Sorry, guys. All right. I drew a paper version of that, or I drew it with my notes. It looks a little bit messy, but I have another one that I kind of was practicing. I'll upload it on top. But does that make sense to you guys? Let me get some thumbs up or thumbs downs on this. I got two. All right, cool. All right, perfect. All right, so again, for the midterm, I'm gonna be wanting something like that, kind of like we talked about now, and I will ask it to you in either way, right? Either 2 genes from 2 different cell types, only found in one cell, or how 1 gene would behave in 2 different cell types. So when you tell me the story of why that gene is on or why that gene is off, I expect you to go from, I want you to just imagine that the DNA is already coiled up, all the DNA is put away, like you just got out of cell division. And once you popped out of cell division, it's your time to, hey, what genes are we going to turn on? And that's where you got to bring in, hey, pioneering factor is going to show up. The only one you need to know is that histone-acetyltransferase, be like, hey, that transferase is going to come in. We're going to acetylate the histones. By doing so, you're going to release the DNA. Now that the DNA is more open, now your transcription factors can go bind to the promoter sequences. This brings in that basal transcription machinery, like your polymerase. You make the gene, and let's say after that, you're like, for closing, there's a little mechanism above it, but you can, or before it, to kind of set up how to turn it off. But for turning it off, I'm okay with just knowing that there's going to be your histo deacetyl, the H stacks are going to be brought in. They're going to take away the acetyl groups. Once you take away the acetyl groups, your DNA is going to coil tightly again, and you basically stored it away. And if you give me that, if you know that whole story from top to bottom, you should be good for the midterm. All right, I think I hit the big questions that I had from the cards. Let me see. How much do we need to know about the enhancers, that binds to between histones. So there again, the enhancers is a sequence where the transcription factor binds. The big important thing there is that normally the enhancer sequences, when the DNA is compact, they're not visible and they're not, you can't use them. The transcription factor cannot bind to them. Once the pioneering factor has unraveled the DNA, then it opens up those enhancer regions. Now your transcription factors can come in and start binding the DNA. How do pioneering factors differ from promoter sequences? And from that one, I believe that's what we're going over right now, which is that the pioneering factors, they come on early on. And the big difference is pioneering factors can grab onto the DNA despite being bound into a histone, while the other ones, transcription factors and the polymerase, cannot bind the DNA if it's wrapped around a histone. All right. Do we need to know specific enhancers on DNA? For this one, you do not need to know it like kind of how we'll be doing with the other stuff. I'm like, hey, memorize ATG, memorize TTA for your promoter sequence. For the enhancers, you won't need to memorize exact sequences. All you'll need to know is that each cell type that's trying to express a specific gene set, they'll share similar enhancer sequences. And then genes that are expressed in different tissues normally have different enhancer sequences. As long as you know it abstractly like that, you should be all right. All right, same thing. Do we need an enhancer sequence? Nope. Okay, I think I answered that one. And during transcription, because where methylation attaches. Perfect. So the histone. Yeah, so here, this is a good question. It's talking about how does methylation fit into the transcription? There, it works the same way, kind of how I went with over acetylation, right? To be honest, we can think about it as all three marks. We have our phosphorylation, our acetylation, and our methylation. Depending which way you're trying to go, right? If you're trying to turn a gene on, you're going to get more acetylation marks and more phosphorylation marks, which are going to release the DNA from that histone, help you turn the gene on. On the other case, if you end up adding methylations, specifically trimethylations, so you're putting 3 methyl groups onto that group, you're going to get the DNA to coil more tightly, and that's going to push to turning the gene off. All right, so acetylation, phosphorylation equals loose DNA equals gene on. Methylation equals tight binding, gene off. As long as you know those, you'll be all right. But the main one is that H-stack and the hat, know them by name. Feel really comfortable with those guys. All right, how does transcription change into... Sorry, my dog's a little crazy right now. How does transcription change in different enhancer groups to determine what is it working with? How does transcription change in different? Yeah, so there, again, this is bringing again to that big story that when we say enhancers, there's like so many different types of enhancers, right? Each cell type can have different enhancers. One of the ones that I used to work with before was a pain in the butt that it was like this gene to turn itself on, it actually had like 4 different types of enhancers. And what it was doing was it was getting information from 4 different cell types. And basically the 4 different genes told it where to make it. And it was like one gene was diffusing this way, one gene was diffusing this way, one gene was diffusing like that, and one was all over the place. And because where all 4 of them overlapped was only in like this little, like it looked like a little bit like this kind of a shape, we could get that specific pattern of expression only because those 4 enhancers had to get together to get a good expression level. If you went above it, you were missing this bottom one, it didn't express. If you were missing this gene, it wouldn't express going that way, it would die off this way. And going this way, you wouldn't have enough of these 2, so it would die off. So it's like the enhancers are made, they're like super complex. They can, for midterm stuff, I'm gonna keep it simple and be like 1 enhancer at a time, and it's gonna be just one like theoretical tissue specific one. In reality, they can be super complex. You can have like I said, multiple different types of enhancers that work together and add up. And you can use that to get multiple positional cues. Like that's the way my system did it. It actually keeps the stem cells happy only if it has the positional cues that, like imagine my stuff was at the very tip of the plants, like imagine at the very tip over here. It had to receive signals from the roots telling it that there's enough water and there's nutrients coming in. There was a signal that migrated up within the left and right where they're making the new leaves. They signal to each other to make the stem cells turn into a different organ. So those signals basically push each other off. And by all those things interacting, I get like this little like triangle shaped cone of stem cells that are maintained based on all those positional cues. So they get pretty complex. They can also have inhibitory effects. The enhancer sequences can turn genes on, which is the main thing I'm going to be talking about in for the midterms, but they can also turn genes off. So you can actually get zones where you have something that tries turning it on. It'll be at a gradient going this way, and you have another gene that wants to turn it off going this way, and it will basically let the gene be turned on to here. Once it reaches a certain level, the gene basically just drops off, and you won't find any more expression coming from it. But that's like across multiple cells. So if you guys ever want to get into that, next quarter I'm teaching, I think it's like advanced molecular technique, advanced molecular, and that one I'm gonna go super detailed about that. But for the midterm, keep it simple, just one type of enhancer sequence for a specific kidney or specific lung, and that's it. Sorry, I didn't go too much into tangent off of that one. All right, let me see. What's the pioneering factors? Can you go over the 4 strategies again? All right, I think that's the one I went over right now about the pioneering factor. The main emphasis is going to be 2 different cell types, and I'll talk about those 2 types of enhancers. And the strategy is going to be this histotransferases and deacetylases to kind of get them to turn on off. Our test is next week. Is it? Sorry, guys. There's no schedule. I think we still got time. Oh yeah, it is next week. It's on Wednesday. What? Okay, yeah, you're right. It is next week. Yeah, so for that, tomorrow on Friday, I'm just going to go over regulation of the second part for regulating cell division. It's going to be like CDKs, but then we're going to include everything coming from after test 1. So it's going to be a lot of the transcription things. Do you guys remember when I went over that section? I do want you guys to feel comfortable with the getting me a protein to a specific compartment. So remember there, my favorites are the nucleus, the mitochondria, and the ER. Because they have the cytoplasmic ribosomes to get the protein to those 3 compartments. At least know the way to the nucleus. The other 2 are very similar, but make sure you feel very comfortable with the nucleus. On the other side is we either have the protein going through the ER, going through vesicles, and that normally gets it to us to the lysosomes, to the surface, or expels it from the cell. Feel comfortable with those, because those are still going to be the big questions I'm going to bring up. I am probably going to make them a little bit more front heavy, because I'm going to ask a little bit more too also about like, hey, how do you think the histones are behaving in order to turn these genes on or off? So hopefully you guys feel comfortable with the things we talked about today. Here. All right, I think we still have a few more minutes. Any other big questions you guys have? Or stuff that you guys want me to go over? For that one. You guys remember I had that one of the slides I had like a really nice picture? Okay, this is a pretty good example of giving us the whole story, and it goes into the we'll talk about it as in the free ribosome, right? So here's the kind of things we were talking about today. This top part about how the chromatin being open or closed is going to determine whether we can even start transcription, right? Once we get into transcription, I do want you guys to feel comfortable with the three modifications. If we're talking about eukaryotes, right, which is your 5' cap, cutting out those introns, and then getting your poly A tail. That's going to give us to this mature RNA, which will be the shorter version. Within that, you're going to find your ATG, which tells us this is where you start making your methionine. That'll be near the 5' side. Start making your proteins based on 3 codons, and eventually you hit your stop sequence, and that's when you stop making the amino acid chain, and it releases it. The only thing's going to happen is that here your mature mRNA is going to get kicked out of the nucleus. This is going to go into the cytoplasm. In the old way, the thing we had talked about the scopes before was the ribosomes were actually glued onto the surface of the endoplasmic reticulum, so the ER, and the mRNA was basically running through that ER through that ribosome. At the same time, on the inside, they were pumping in the amino acids, right? The free ones, the other hand, is they're literally just floating around in the cytoplasm, right? So they're outside of the nucleus, outside of the ER, just literally floating in the cytoplasm. You have your mRNA, the ribosome will jump on, and it'll start doing the same thing. It'll scan, find the ATG. Then the small subunit scans, finds the mRNA, finds the ATG. You bring in your tRNA with your methionine. You bring in your big subunit. That makes the big complex for the ribosome. This complex then starts to move across this space like that, doing every 3 codons, adding another amino acid. On top, you're linking up your tRNAs. Sorry, guys, give me a second. Sorry, guys. So at that point, you have your ribosome making your protein, but the protein that's getting created is literally still just floating in the cytoplasm. Because it's floating in the cytoplasm, it's not inside of a vesicle. He's going to be in trouble. Hey, it's not in a vesicle. You have a free floating protein. That free floating protein is literally just free in the cytoplasm. So the cool part about this is that in order to get transported into one of those compartments, which is the nucleus, the ER, and the chloroplast in plants for photosynthesis, is that all these 3 organs have those double membranes, right? So it's like we can't just throw protein like a vesicle into it because it'll get put inside of the double walled, not all the way through it. So these get transported. So most of these have like different little tags, which is the amino acid sequence has a little sequence part of it, like the NLS sequence. It has like a leucine, a spacer, a leucine, a spacer, and another leucine. So if you put 3 L's in a row, skipping one, that sequence tells it gets recognized by a transcript by a factor, and it gets pushed into the nuclear pore complex. Actually, I think that's the slides that I wanted to see. Let's see, show me what this is. V6. I'm gonna have to look that picture up for you guys because I feel like I put it in my slides. There it is. Look at this gorgeous picture. Yeah. So this is one of them, right? So the mRNA, and as you can see, the ribosome is literally just sitting in the cytoplasm, making the protein in the cytoplasm itself. And then that beautiful picture. Didn't put a single picture of it. Let's see. All right, I'm gonna show you one really fancy picture just because it looks so cool. Nuclear or complex NLS sequence. Mm-hmm. So here, if we look at this picture on the right, the ribosome sitting in the cytoplasm just finished making the protein, and it's a chain, right? The methyl group in the front, you have, methionine, then you have all your amino acids. Part of those amino acids, there'll be a sequence of them that'll function as that nuclear localization signal. So that'll be something that gets recognized based on its like charge, the amino acids you have there, and what it can bind with. This normally happens to be these additional proteins, which is called like importance. Those will go and recognize that exact sequence here, kind of shown in orange. When they bind to that, they actually make this bigger complex. This gets kind of trapped together on that protein. Like say my hand's the protein, this is the tag. It recognizes it, it brings it into this nuclear pore complex. This thing's like a little basket. It has like little fingers that grabs onto proteins and basically pushes them into the nucleus. But it physically interacts with them, grabs onto them, brings them in. If they don't have the nuclear localization sequence properly fit into this important complex, it won't let proteins in. So that's like the bouncers that are protecting the nucleus, right? If the whole thing is complete, it will grab onto it and it'll import it into the nucleus. Now you have that protein kind of showing up on the inside. In here, you actually release those factors that had grabbed onto that nuclear localization signal. They basically let go, and now you release the protein. In some cases, they cut it off, but for the most part, they can just let that protein go. And now you have that protein that was made in the cytoplasm. It gets moved through that nuclear pore complex, and now you'll find it inside of the nucleus itself. So the nucleus does it the same way. What's the other one? The mitochondria does it, and the chloroplast does it. So look, if we check it out like this. Yeah, I do remember talking about this. You guys remember this one? Tick and talk. So it's doing it the same way, right? So here's your free ribosome. In black, you have your protein. In yellow, you have that part of the protein that serves as a recognition sequence, right? This interacts with the talk complex. They basically like, oh, there's your tag, grabs onto it, brings it into this talk tick, which will be these two proteins that sit on the plasma membrane. So the phospholipid bilayer, this light green area would be one, this dark green area, the black line there will be the second phospholipid bilayer. These will then transport the protein through both of those. So you get past the bilayer, and then boom, you can put the protein on the inside. In this case, you can see here, they cleave off that yellow part, which is the tag, and you get left behind with just the black part, which is the actual protein that you care about. Yeah, here's a super zoomed in version of the process. Here in black, you can see that this is the amino acid sequence you're trying to push in through the complex. Here you see that this yellow and green would be this outer phospholipid bilayer, how you're getting through that bilayer. Here's your phospholipid stuff, the outer membrane. Here's your inner membrane. And here you can see your tick is made of multiple proteins that help get it through the other bilayer. And just like that, if we also look it up, I think what did I say was the other one? The mitochondria. See, you're going to see the same thing. Yeah, Tim Tom. That's the other one. I remember this one. Tim Tom, again, ribosome sitting free in the cytoplasm. Your mRNA gets translated into this protein. The protein then gets pushed through the Tom complex, then through the Tim complex, and then boom, you have your protein all the way on the inside of your ribosome. Sorry, mitochondria. So if you can explain it for the nucleus, you should be comfortable with the mitochondria and the chloroplast. I don't need you guys to know these names of Tim, Tom. Just know like, hey, there's proteins that can kind of help get our protein across both phospholipid bilayers all the way to the inside. If you remember the nuclear pore complex, great. I appreciate the notes, but as long as you say, hey, there's a protein that transports it, I'll be fine. And then for the other pathway, remember the proteins are on the ribosomes. Sorry, the ribosomes are on the ER. The protein gets made inside of the ER. And then you have vesicles that get moved from the ER to the Golgi. The Golgi does these extra modification steps. At the other side of the Golgi, you go from the cis trans to the trans side of the Golgi. At that point, the vesicle gets guided either to the plasma membrane to dump things out or to the lysosome to kind of start digesting things. And what's the other one? Yeah. So here, I remember it's nucleus, mitochondria, chloroplast will be the ones I care about. On this side, it'll be plasma membrane, secretory vesicles, and the endosomes. All right. Any other questions? I lost the chat. Okay.
Thank you.
Thank you. Hold on, I'm trying to figure out the mutation ones. I think if you guys are good after this, I'm gonna let you guys go. So the mutations, remember the first three types when it comes to if a base pair changes, know the difference between silent, nonsense, and missense, right? So nonsense is stop, missense is it switches amino acids, and then silent means nothing happens at the amino acid level. And then for the big insertion deletions, the only two I'm going to be picky about is does it cause a frame shift or not? So I might be tricky and be like, hey, I'm going to have you guys make me an amino acid change. I'll insert something or remove something and then just bring it up. Does this cause a frame shift? Yes or no? So go over those guys. If not, we're going to have, I think, one more review next Monday. And we'll be doing, I think this Friday, we have the second part to cell division, and you guys will have homework on Friday. All right. And I think if no other questions, I'll probably let you guys go. Give it a, you guys good? All right, give me a thumbs up if you guys are good to go. Thank you guys.
Thank you.
Thank you guys. All right, see you guys on. Oh yeah, see you guys on Friday.